Hello and welcome to the eOrganic webinar on grafting for disease management in organic tomato production, presented by Frank Lowes of North Carolina State University and Carrie Rivard of Kansas State University. In this webinar, you'll learn about tomato grafting and how it can be used to manage diseases in organic open field and high tunnel systems. The presenters will provide information regarding rootstock selection as well as the grafting procedure. This is eOrganic's second webinar on tomato grafting. David Francis of Ohio State University presented a webinar last year, which can be viewed on our website under archived webinars at the address on your screen. This webinar will also be recorded and will be available there within the coming week. My name is Alice Formiga, and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. In addition to all of our recorded webinars, you can find information about upcoming webinars as well as articles and videos on organic farming on our website at extension.org/organic_production. We're very excited to have Frank Lowes and Carrie Rivard with us today. Dr. Frank Lowes is a professor of plant pathology and the director of the NSF Center for Integrated Pest Management. He enjoys advancing the science and practice of growing vegetables and training others to do the same. Dr. Carrie Rivard recently joined the Department of Horticulture at Kansas State University as the Fruit and Vegetable Extension Specialist. His research focuses on grafting and high tunnel production for organic and conventional growers. So now, without further ado, I'm going to hand the presentation over to Frank Lowes.
Thank you very much, Alice, and welcome to all our guests. We're very pleased to do this webinar together, especially to do it together with Carrie Rivard. Carrie Rivard did a lot of work here in North Carolina, but he just finished his PhD and got a faculty position there at Kansas State University. So we're very proud of him that he got that type of position. So this is a picture that shows some of our grafting teams, a few members of our team. Of course, Dr. Mary Pete, many of you know, and she is now the National Program Leader for the Organics Program at USDA. In their USDA program. Steve Moore is a high tunnel guru, and so he's done a lot of work with high tunnels, and he helped us a lot with their grafting program. Susie O'Connell is a PhD student that she just finished up also and did a lot of work in tunnel systems and field systems with grafting of tomatoes. What I wanted to highlight, here's a picture of a grafted tomato, and we see here we have the rootstock. We have the healed area where the graft occurred, and then we have the Scion. I often refer to the Scion as YFT, or your favorite tomato. And you can put your favorite tomato on a rootstock as long as they're compatible. What we'd like to do today is we'd like to first provide some background about tomato grafting, and then we'd like to show some data where grafting has helped a number of organic growers in our area. And then we'd also like to demonstrate how actually to do grafting. And then, of course, we'll have that question at the end. So grafting was first reported in vegetables in the 1920s to manage Fusarium wilt of melon. And then since that time, it has rapidly expanded, where 81% of the Korean industry and 54% of the Japanese vegetable industry uses grafted plants. And likewise, many countries in Europe and other places use grafted tomato plants in the majority of their production systems. These are some photos that Mary Pete had taken while she was in Asia. When we do grafting, what we're trying to do is we're trying to take advantage of the root system. This picture actually shows where we have a grafted plant and we pinched out the leader, the apical meristem, and then now we're training 2 leaders off this plant. And so what we look for in a root system is biotic tolerance or abiotic tolerance. And by biotic tolerance, I would mean disease resistance to a lot of soil-borne pathogens. Abiotic tolerance would be tolerance to extreme pHs or high salt conditions or other extreme soil conditions. And what we look for in a root system is enhanced water and nutrient uptake and efficiency and nutrient assimilation. And also we can envision that now that we've delinked the roots from your favorite tomato, the question arises, can we breed rootstock that are better suited for organic systems to take advantage of the microbial communities that are in those systems? And so that's certainly an area for future research and practice. Most of our work has occurred in North Carolina. North Carolina is a long state. We have the mountain region in the west, and we have the coastal plain in the east part of the state. We have very diverse types of diseases, and mostly in our work we have focused on the major soil-borne diseases in North Carolina, including Verticillium wilt, Fusarium wilt, the root knot nematode, southern blight, and bacterial wilt. And a lot of our research was done on research stations, but also on farms. And we really highly value on-farm research. And here's a slide that shows some of the folks that we've worked with, Alex Hitt, Ken Dawson, and Stephen Hartman. And the reason we highly value on-farm research is because I think to move any new technology or method, you need to closely link the practice and the science. These growers are great practitioners, and of course, part of our role is to advance the science, and by linking together, you can really advance the farming systems. Any new technology, like grafting, it needs to be put in within the context of a farming systems project, as well as on the farm in their farming system. So the practice and the science needs to be closely linked. And it's really enjoyable to do on-farm research because both parties learn a lot. And one thing that we really wanted to highlight, that if you're going to use grafting, it's going to be very important to understand what the problems are in your soil. I'm going to highlight these 5 major soil-borne diseases. And the rootstock selection is highly dependent on the site-specific problems that a grower has, and or what the objectives are for the grower. And so no rootstock suits all farmers. Let me start with bacterial wilt. Bacterial wilt is caused by a bacteria called Ralstonia solanacearum. It's a true southern disease. You won't find it in the northern production regions. But this bacteria has a wide host range, including most of the solanaceous crops like tobacco and potato, eggplant. It's a very diverse pathogen, and there's been a lot of work in tomatoes, and there is some good resistance in tomatoes. And what I wanted to highlight in this slide, this is a work that we did at Stephen Hartman's. Here on the left, you can see the bacterial wilt. We have a scale from 0 to 100%, and if we do not graft, by the end of the season, 100% of the plants die. Now in all our slides, or most of them, we have self-grafted plants, and we just do that for scientific purposes. We take our standard variety, we cut it, and we graft it to itself, just to take into account the grafting effect. But look at this DP105, it's also called RST04105 from DP Palmer Seeds. And you can see that it gave 100% control of bacterial wilt on that farm. So we went from 100% disease to 100% control, and that essentially doubled the yields. We went from about 10 pounds per plant up to 20 pounds per plant. So in our experience, we've used open breeding or open lines. They're breeding lines. So growers could actually grow the seed themselves on the farm, and as long as they use good sanitation practices. But CRA 66 and Hawaii 7996 have performed well in our bacterial wilt trials. And then we have a couple others, Don Hanmei and the RST 105, that also have performed well. Here's another slide showing how the bacterial wilt has performed for us. You can see here there's a plant. We get the sudden wilt due to the bacteria. It colonizes the plumbing system of the plant. It often you get sudden collapse when first fruit set occurs. Here you can see a field where many of the susceptible plants have complete wilt. And then we have some resistant lines that are not wilting. And so it's very dramatic, the type of loss that one can experience. So in this graph, this is another study. You can see if we don't graft, we have 100% plant wilt. And the 105 line, we got 100% plant stand. Now this Hanmei, that's actually very interesting because it performs really well in our mountain production region. And then here in the east, we get some wilt. And 105 shows the opposite effects. And so this has to do with the genetic diversity of the pathogen. Some tomato lines are better against certain strains, and other tomato lines are better against yet other strains. And so rootstock selection becomes critical even for controlling specific strains of a pathogen. The next disease we wanted to talk about is Fusarium wilt. This pathogen is very common throughout North America. It's a fungal pathogen that persists in the soil for years, and it gets into the plumbing system causing wilt. And often you'll get what's called a unilateral wilting. You can see these yellowing leaves because the plumbing system on this side of the plant is plugged up. And there are 3 different races that we know of, race 1, 2, and 3. And a lot of commercial tomato lines in rootstocks will have resistance to 1 or all 3 of these races. So this just shows some data. This is from Alex Hitt's farm. We can see that if we don't graft, we have up to 50% plant wilt. And here Maxifort has excellent resistance to this particular strain of the pathogen. And of course that will impact yield. The next pathogen we wanted to highlight is root knot nematodes. And this is a southern root knot nematode, but there's also Melodogyne hapla. But the nematode penetrates the roots, and you can see here, this tomato root, we have 100% galling, severe galling of the root. A lot of rootstocks will have what's called the MI gene, which does give resistance to the nematode. But that resistance, it seems like it varies with the background that it's in. This particular study is not on an organic farm. But you see, if we don't fumigate, if we don't graft, and we have a susceptible line, we get 100% root galling. That's a rating of 10, and that's like this picture here. And then if we graft with Beaufort and Maxifort, we do get some root galling towards the end of the season. This is under a plastic culture system, so we may have encountered very high temperatures, and that MI gene is known to break down in temperatures, in high temperatures. And so we would call these 2 moderately resistant to this particular strain. But look at this one here. This is Big Power. It had no galling even after 90 days. And what was very impressive is that at the end of the season, the plots from Big Power only had about 40 to 49 nematodes per 500 cc's of soil, whereas all of these other plots had anywhere from 1,200 to 2,500 cc's of nematodes. And so it's very impressive how this rootstock suppressed the disease, but also suppressed the multiplication of the pathogen. Southern stem blight, again, it's a southern disease. You won't find it in more northern production regions. It's caused by Sclerotium rosei. It lives in the soil. It has a wide host range. You can see how the pathogen, it lives in the soil, and then the plant emits chemicals which causes these round structures called sclerotia to germinate, and then the pathogen colonizes the base of the plant, and then you'll get complete wilt of the plant. Now, Carrie and I were very surprised with the data that came in, and it was, I wouldn't say it by luck, it was serendipity. I don't believe in luck too much myself, but there's no resistance in commercial cultivars. But when we put rootstocks on an organic farm as well as in one of our research stations, we found that the non-grafted plants had up to 80% plant wilt, but our grafted plants had no wilt at all. So it offered very superior, and we would call it tolerance. We don't know that there's resistance per se, but we do see tolerance. We've also done work with David Francis, and some of those Ohio breeding lines also have a similar level of tolerance where we're able to manage the disease with those grafted plants. So again, we see very dramatic differences between non-grafted and grafted plants under disease pressure. The next set of slides, these were done in Pennsylvania at Cedar Meadow Farms with grower cooperator Steve Groff. Now Steve grows most of his tomatoes under these hay grove high tunnels. You can see he has a whole series of them. And one of his main problems has been Verticillium wilt. And this is caused by Verticillium gallii, and it lives in the soil, and again, it colonizes the plumbing system of the plant, and then you get wilting of the plant. You can see this top of the plant starting to wilt, and then you get these lesions on the leaves, and the pathogen also is colonizing in the plumbing system producing mycosclerotia. This is a bit of a challenge to manage because we know that there's two races, and most tomato lines that are hybrids will have race 1 resistance. Now most heirlooms won't. So a rootstock could offer resistance to a portion of the population, which we would call race 1. But what we found is that a lot of our rootstocks seem to give a lot of vigor to the plant. We have a large root system, and this seems to offer a tolerance to Verticillium wilt. And Steve Groff saw that at his place. But what we wanted to highlight at Steve's place was he asked the question, how can I reduce the cost of my rootstock plants? And what he was finding is some of his rootstocks gave too much plant vigor. Normally, if he did not graft, Steve would plant his plants at 18 inches between the plants. So we did a test with him, and we looked at plants planted at 18, 24, and 36 inches. And what this graph highlights is that we can take Maxifort rootstock and train 2 liters, and what we found is that you can have the same yield at half the plant density as standard plants planted at 18 inches. And I think Steve is ending up somewhere around the 24 inches between plants because that's where he gets his maximum productivity and maximum profit. But that's a dynamic process. I know he's often trying out different things. So this table summarizes a lot of the experience that we have in North Carolina, and looks like the bottom portion of that slide got changed a little bit. But what it highlights, hopefully, we have a handout where this table is very clear. This table got a little crunched up in this setting. But what we have here is we have rootstocks, and based on our experience, or based on releases from the seed companies, we have indicated what kind of resistance these lines may have. For example, we see that all of these rootstock have excellent resistance to Fusarium race 1 and excellent resistance to Fusarium race 2, except for this dihomai. All of the lines have resistance to race 1. They all have the VE gene, but they do not have resistance to race 2, and as indicated, we're probably looking at a tolerance based on our work. We showed that root knot nematode, probably Beaufort and Maxifort, offer moderate resistance, whereas Big Power, which is on the bottom here, we saw that offered very superior resistance. Likewise, we saw dihomai and RST 105 give high resistance to bacterial wilt, depending on the geographic site that rootstock was used. And again, for southern blight, we see high resistance or modest resistance. And so these tables are available. We just wrote a review article where we've compiled a lot more rootstocks, but this table highlights our North Carolina experience. Finally, we wanted to highlight some work that we've done in high tunnels at the Center for Environmental Farming Systems. Here's our high tunnel systems, and we compared outdoor production versus indoor production, various planting dates, various nutrient levels, and different types of rootstocks. And I'm just going to highlight the rootstock work. Here you can see the field production system where we have a rootstock and the Scion, highly productive and non-grafted. Here's the tomatoes growing in a high tunnel system, and we had cover cropping systems outdoors to encourage beneficial insect populations. We were running into some insect problems, and so we increased the beneficial populations. So this is data from 2 years of work, and they both show very similar trends that in these high tunnel systems and in the field systems, we had no obvious disease pressure. Because a lot of growers will ask us, well, should I graft if I don't have any known disease pressure? And in this case, you can see in the open field in the high tunnel system, Maxifort increased the yield by 42 to 53%, and likewise, just taking another year, where Beaufort did a little bit better, In the open field and the high tunnel, we increased yields by 35 to 37% as compared to the non-grafted controls. So we do see that in both the field system and the high tunnel system, we did get a benefit from these vigorous rootstocks, even in the absence of disease pressure. Now, I wanted to highlight work that other people are doing in the U.S. There's a number of programs, and of course, I'm not presenting an exhaustive list, but Carol Miles and Sasha Johnson in Washington State University, they're doing work on looking at grafting for managing Verticillium wilt. Again, it's a temperate climate pathogen. They're also looking at the effect of grafting on fruit quality, yield, and plant vigor, and they're doing a lot of work trying to optimize the healing chambers, especially for the organic grower community and smaller growers, smaller acreage growers. There's a lot of work that's being done in Florida. Dr. Xin Zhao, and we talked about how these rootstocks might offer enhanced nutrient as well as water efficiency. So she's really looking at that question. Can we reduce the water load? Can we reduce the nutrient load? And I think this is very important for organic growers. Depending on your nutrient source and how you manage the nutrients, you know, if you can have a rootstock that's better at mining the nutrients that are in the soil, then you can enhance the productivity of your land. And she's also doing a lot of work with sensory and fruit quality impacts like post-harvest, and a number of programs do look at that. In Ohio, they've done a lot of work, and Matt Kleinhens is a horticulturalist, David Francis is a plant breeder, Brian McSpadden Gardner and Sally Miller are plant pathologists. And this is just some data from them. And I put this up just to contrast some of the data that we had here in the South. In some cases, you can see this is yield over time. And these are hybrids that Dr. Francis is developing, and we've had the privilege to work with him and evaluate them. And I know his webinar was February 2nd in 2010, and Alice indicated you can go see that. But you can see in 2008, those hybrid rootstocks enhanced yield. In 2009, there was not too much difference. And in 2010, we saw that the 3 program hybrids did not do as well. So in those cases, there's not as dramatic of an impact between the rootstock and the non-grafted plants. And we also see that it varies from year to year. So I think this is where the practice and the science comes together. As the organic grower community works with grafted tomatoes, we will learn more and more where these rootstocks will have an important role for our growers. In the next slide, this is the Ohio team has also put together a pictorial guide for using the cleft graft method, which is a different method of grafting. So that's available. And finally, there's a program in Arizona. Sherry Kubota has a really nice grafting website, and she does a lot of work with grafting, particularly with melons, and also trying to optimize grafting. And then there's a USDA group in Florida, Nancy Callis-Burrell, Aaron Roscoff, Michael Bosher, and they're in Fort Pierce. And they also do a lot of work with tomato grafting in open field systems. So in conclusion, by way of background, here you can see our high tunnel system. What we did here is we trimmed the central growing point, and then we're training 2 leaders, so we can have half the number of plants per acre, but still the same number of stems in that area. So what we find is that grafting provides a site-specific management tool for many soil-borne diseases. And I highlighted the need to make sure that you have a good understanding of what the problems are. And then we saw that the rootstocks can increase yield through plant vigor or through enhanced water or nutrient efficiency. And there's opportunity once you adopt certain rootstocks, maybe to change the plant density, to change the fertility levels, or to change the training practices. And so we can integrate those into the farming system as you practice it. And I'm going to turn the slide over now to Carrie Rivard. And
can you guys hear me okay?
You're on, Carrie, yeah.
All right, Alice, do I have control of the slide? I think I do. Okay, so I'm going to real quickly just give you a quick rundown of how we actually carry out the grafting at NC State. Most of our grafted plants we produce for our research trials were produced on campus, and we grew grafted plants in a range of environments, from a typical large, more commercial-style greenhouse to indoor growth chambers as well. Here you can see a picture of Susie during one of our late-night grafting events. The other thing I would suggest is point your direction towards this extension publication that Frank and I put together a few years ago. This has been available on the NC State website as well as the Ohio State website, as well as others. And what's nice about it is it gives sort of a step-by-step, this is how you carry out the grafting process. Now what I'm going to do today is basically try and fill in some of the gaps that article does not. So I'll try not to be very redundant between that article and what we'll talk about. And the other thing that I would note to go along with that theme is that there's really no recipe for success whenever you're putting together a grafted propagation system. Keep in mind these are biological organisms. They're going to behave a little bit differently in your greenhouse than they do in ours, and so there's going to be some variation in terms of germination rate, growth rate, things like that. And so you're going to have to allow for some flexibility within your system when you start putting this together. Now, the other thing that I like to point out is when you start putting together a grafted transplant production system, rather than getting really caught up on the details of the slicing and dicing, instead it's good to think about some general principles of plant propagation that are really important to ensure your success at this. Now, one of these is that you have to be able to produce a large number of numbers, so production is key. Finding a high throughput system like the tube grafting method is really helpful. The other thing is you really want your transplants to be nice and uniform. Keep in mind that on the day you graft your plants, you basically want the rootstock and the Scion to have the same stem size. So what this means is that they have to be very uniform across the rootstock from the Scion, as well as uniform within the crop. The other thing that becomes really important is once you've severed these plants from their root systems, you're going to have to learn how to deal with water stress. And managing that healing chamber is very difficult. Again, a lot of people get excited about the actual cutting of the plants, but one of the hardest things to manage when you start implementing these systems is actually managing that healing chamber and keeping it from turning into an oven while making it the proper environment for success. The other thing that we can't reiterate enough is the importance of sanitation in this process. Bear in mind that you're going to be putting these plants into a healing chamber where we raise the humidity and cut down the light, and this is an excellent environment for disease to break out. So you want to make sure that your clips are very clean, your cutting blades are clean, your hands are clean, and make sure that you're using proper sanitation methods to disinfect everything that you can before you go through the process. And also, it's really important to reacclimate the plants afterwards and make sure that they've hardened off good before you put them outside. We've never had any problems really with plants failing at the graft union due to wind or anything like that, but we're also really careful to put them in the greenhouse for a week and then put them out into a cold frame for another week to make sure that they've hardened off well before we put them out into the elements. Now, the other thing that it's important to keep in mind is that grafting two plants together is obviously going to add a fair amount of cost. And so as you're designing a grafted transplant production system, it's important to remember where these added costs come from. Now, Frank and I, as well as Susie and Dr. Pete and others, put out this article in Port Technology last summer. And I'm not going to talk about any of the details in it, but one thing that's nice about it is that it shows you where some of these added costs come from. And again, some of it does come from the grafting labor. You can see here 14 to 24% in this case range from the grafting labor, but it's also other factors as well, such as the cost of the rootstock seed is very expensive. And bear in mind that you have to grow a Scion crop or a rootstock crop, and this takes twice as much space in the greenhouse, twice as much soil, twice as much plastic, so on and so forth. And so those add costs as well. Now, we do utilize the tube grafting technique. This is the technique that's most utilized by the big commercial operators if you go across the pond over to Europe or even up in Canada. And one of the things that's nice about it is it's a very high throughput system. Basically, the plants are grafted when they're very small. Basically, they're seedlings with 2 to 4 leaves. And you can see the silicone tube basically holds together the Scion and the rootstock while the graft union forms. Now, this is a diagram that comes out of that extension publication that I mentioned earlier, and I'm not going to walk through step by step what happens, but essentially it's important to keep in mind that the plants will be, you may have to stagger the seeding dates if the rootstock and the Scion grow faster than each other. Again, this uniformity of rootstock and Scion crop is very important in the tube grafting method. So sometimes we stagger our seeding, whether we put the rootstock ahead of the Scion or vice versa. It really depends on the Scion cultivar or the rootstock cultivar. And then we typically graft the plants when they're about 3 weeks old, and we put them straight into that healing chamber where we manipulate the relative humidity as well as the light to basically offset the need for that root system, and I'll talk about that here in a few minutes. And we typically say that it adds about probably a week to a week and a half onto a non-grafted production system in terms of how long it takes to grow the transplants. So if it takes you 5 weeks to grow a non-grafted transplant, it's going to take more about 6.5 to 7 weeks to grow a grafted one. Again, the most important thing when it comes to seedling and transplant production is the uniformity of the crop. So you may have to stagger your seedling dates a little bit in order to get that rootstock and the Scion to be the same size on the grafting day, but then also take additional steps to ensure uniformity. So I get a lot of questions about direct seeding versus sowing, versus transplanting. And so one of the things that we like to do is we do like to basically sow into small seedling flats and then transplant out into our plug trays. And one thing that this does is it allows us to select plants that are more uniform. We're able to escape some of those guys that are really large or really small. And it also sort of slows everybody down for a couple of days so they can catch up and again make a more uniform transplant crop. Now in terms of numbers, we usually have very good success. We've had some catastrophe batches, of course, but in general we get above 90%, and we've had several batches where we haven't lost a plant. So this is about the size of the tomato plant when it's grafted. You can see it has about 2 to 4 leaves. Now the actual silicone clip comes in 2 different sizes, either a 1.5 or a 2 millimeter diameter on that clip. We like to use the 2 millimeter clips, and they fit onto a plant about this size, nice and snug, just snug enough to hold it, but not so that it damages the plant. Keep in mind that if you do have a rootstock crop or a Scion crop that's getting away from you and it's growing too fast, that you can use temperature to slow the crop down if it's getting a little bit too large for the other one. It's important when you're carrying out the grafting procedure to really treat this like you're going into surgery. One of the first mistakes I made was I figured out, this is not the kind of thing that you want to go out into the greenhouse at 3 in the afternoon and do to a plant, because at 3 o'clock in the afternoon on a full sunny day, the plant is transpiring very quickly and is generally under some water stress already. So one of the first things that we do is we actually take the plants out of the greenhouse. We'll put them in a head house or some other shaded area where they can basically slow down their transpiration stream and relax a little bit. The other thing that I like to do is actually do the grafting process at night, and that's another good way to make sure that the plants aren't water stressed before you put them through the procedure. The next thing to keep in mind is that you want to absolutely have a nice clean working area. Now this doesn't mean that you have to have a laminar flow hood or anything like that, but just make sure that your tools and your clips and your surface area is very clean. And basically I like to use alcohol to clean stuff, or you can use bleach as long as you rinse it very well at a 10% solution. I like to carry out the grafting indoors because it really reduces the stress on the plants. Basically, how much water stress is on the plant when you begin this procedure is going to dictate how long it takes before that Scion starts wilting. So it's nice to have the plants indoors, have them nice and relaxed and ready to go. And then it's also really important to have your healing chamber up and running and make sure your relative humidity is nice and high before you start cutting any plants apart. So here you can basically see that we cut the rootstock first, of course. I get a lot of questions about the actual angle of the cut. One thing that I would say is I wouldn't get too caught up on the actual angle of the cut. I've never gotten a protractor out or anything like that to check to see what angle we use. I think generally it's about a 45 to a 60 degree angle. But what's really important is that it's consistent between the rootstock and the Scion. And so making sure that those two cuts are nice and consistent is going to make sure that those two plant parts come together very, very snugly within that silicone clip. Now here you can basically see we cut the rootstock And then basically the silicone clip just slides right over top. Now, one thing that I should mention, these pictures came out of that extension publication too, and this is kind of an old article. One of the things that we've learned since this time is that it's better to graft below that cotyledon leaf because you'll have a lot better luck in terms of not getting suckers. If you graft above that cotyledon leaf, you may have a few more suckers, which can be kind of a problem. And then you go ahead and just insert your Scion into that clip, and it should slide into there real snugly. And you can basically see that the angle of that Scion cut is very similar to the angle on the rootstock. And that's what you want it to look like as it goes into the clip. Now, once you've done the actual grafting procedure, the next step is to put it into the healing chamber. Now, while the plant is in the healing chamber, there's a lot of biological processes that have to occur as well. The plant basically has to reform all of its plumbing tissue and reconnect those vascular bundles within the stem. Now, basically what you're doing within the chamber is you're reducing the water stress problem so that it doesn't need that root to survive while it's undergoing this process. And so that's the general idea with managing the chamber. What we do within that chamber is we alter the plant's physical environment so that we can offset the functional effects that the grafting procedure has incurred. In other words, we make it possible for that Scion to survive without its root system for 3 or 4 days while it's healing itself and reforming that graft union. So water moves through the plant. Basically, this process is driven by evaporation of water from the leaves into the outside air in the transpiration stream. So what we do with the healing chamber is we basically put enough humidity into the air that we cause a traffic jam, and we prevent water movement out of the leaf and into the outside air by increasing the humidity and reducing the light and essentially cutting down that transpiration stream. We've built a lot of different chambers on campus at NC State. Many of them utilize these cool mist vaporizers that you see in this picture here. These are available from department stores. You can also get a bigger version of this called a hermetifier from many of your horticultural supply companies. For our purposes, we generally can get by with the smaller vaporizer. One thing that I would note is that make sure that if you're not using a warm water vaporizer to add humidity, and the other thing is not to use a hose-in mister, sort of like a hose-in mister we'd use for watering seedlings. Any kind of physical activity or that physical force that you use if you spray water into that chamber will take the plant and make those Scions fall out of the clips. So you really want to add moisture as a vapor and don't spray it in with those hose-in misters. Here you can see some plants that have been recently put in the chamber, and if you look at the bottom right-hand corner here, you can see plants that were maybe just put in versus ones that have been in for maybe an hour to an hour and a half. And you can see how quickly they pop back up within that chamber. That humidity really pops them right back up. The other thing I might point out, and this picture shows it well, is I like to use these 50-cell plug trays that we cut into strips of 20, and you can see how I've cut those here. And those are just nice because when you're physically working with these things all the time, you don't want to be reaching across the tray to actually get to the clip or to do the grafting. So what's nice about these strips is you can basically work down one side and then flip the thing around and then work down the other side. Here for a home gardener, this is a great way to start. We have had successful batches using some of these biodomes. You're scaling it up a little bit, and basically what you can see with the chamber is that it's a very inexpensive structure. We like to use PVC for a frame, works fairly well, and then of course plastic is basically used to hold in the humidity. You do want to make sure you have a plastic floor as well because the humidity will drop out. But then we basically use black plastic or shade cloth in order to cut the light out. Now one thing I would note is that this camping tarp right here is actually the most important component of this structure because what it's doing is it's keeping the direct sun off of this chamber. Bear in mind that especially when you put the black plastic on, that if the sun's direct rays can hit that chamber, it's going to cook everything inside of that chamber. So you're going to have a nice, good way of blocking the sun's direct rays. This is in one of our indoor chambers, and it's a little bit fancier. It also uses the cool mist vaporizer. The only thing I would note is that you can see we've actually stacked plants in 2 different levels. You don't necessarily need light during the first few days of the process, so it's okay to think vertically when you're building your chamber. Here's one at a grower's farm. This is actually a Steffen's farm, and again, these are very inexpensive. You can see this is a rebar and cinder blocks basically made the construction. And again, the most important component is this weed fabric right here, which is on that southwestern exposure, keeping the direct sun off of that healing chamber. Here we're scaling it up a little bit. This is at a grower's place in Pennsylvania, and basically we use wire hoops as well as the poly film. And in this case, he was actually on an ebb and flood bench, which was really handy. And then you can see he basically has layers of shade cloth across the top of that healing chamber. And so maybe on day 5 you might take a layer off, and day 6 you would take a layer off, day 7 you'd take a layer off, and then maybe day 8 you'd take the last layer off. And that's sort of the way that works. Now if we go back to the picture of Korea that Frank showed earlier, you can basically see all the same components that I've been talking about. The most important one, notice how there's shade cloth across this entire greenhouse. So in this case, they were keeping all the sun out. And then, of course, they basically just use poly to hold in the humidity. Further shade cloth to cut down that transpiration and cut through those sun's rays. Once the plants have been grafted, they're quickly, we put them into a greenhouse for about 7 days or up to 10 days to harden off. Now during this process, the clip itself will actually just open up and fall off the plant on its own, so you don't need to worry about that. You can actually pick those up after. I like to pick them up when I'm planting out in the field, and of course I come home with a big pile of grafting clips, but that's okay. And you can actually reuse those as long as you disinfest them with something like bleach. One thing I would note about overhead watering, of course it's very important to be very careful when you're dealing with these plants in terms of overhead watering. We like to use overhead watering probably on the 3rd day in the healing chamber, maybe on the 4th, and you'll start to use it about every other day within the healing chamber, and then just be real careful with it once you pull them out of that chamber. Once you get them back to the farm, there's a couple other things to keep in mind. One, planting depth is very important. Bear in mind that the upper portion of this plant, the Scion, may be susceptible to all these diseases that Frank was talking about earlier. And so we want to make sure that graft union is as high above the soil as possible. For the same reason, we like to, if you're using a stake and weave system, it's good to get that first string on the plants as quickly as possible to lift them up off the ground. The other thing to bear in mind is that you may see a few suckers come from the rootstocks. And generally, this isn't a big problem. Again, it's important to graft below that cotyledon leaf on the rootstock plant. And also, if you're doing some suckering, we usually catch the suckers when we do all of our other pruning. The other thing just to be aware of is, again, many of these rootstocks are very vigorous. Here you can see a picture of Cherokee Purple grafted on the Maxifort. And again, the size of these plants is something to contend with. And so it's important to bear in mind what you're going to do with those plants. And also, maybe you have to use larger stakes to hold them up, or your trellis system may need to be adjusted. And one of the ways that we've done that is by using this double leader system that Frank mentioned earlier. And here you can basically see it if you're using a string trellis system. It's real easy to adapt that system. In the stake and weave system, we have used it especially for really wide spacings, for like that 36 inches, for example. But for the really close spacing, it may not be necessary because you get enough branching from the plants just on their own. So in review, basically one of the things to really keep in mind is the uniformity is going to make it a lot easier to graft the plant successfully. And it's one of those things that when you're actually carrying out the grafting procedure, it can be either a really fun task with a nice small uniform set of plants, or it can be a really arduous, hard, frustrating task if you have plants that are a little bit too big and are poor uniformity. The other thing to keep in mind is that once it's time to actually carry out the grafting process, timing is critical. And these plants are growing very fast at the time that they're in their lives, in their life stage. And so the difference between 1 to 2 days can be all the difference in the world when it comes to fitting them in those little silicone clips. And then just make sure you're real careful to watch them during the healing chamber period. I like to check on them at least twice a day. and many times I'll schedule things where if I'm going to remove a piece of shade cloth, I'll come back 45 minutes later to make sure that it hasn't heated up too much in that chamber, and so on and so forth. So just keep in mind the activity that's happening within that chamber, and make sure you're very diligent about observing what's going on. And with that, I think we'll go ahead and take some questions. Alice, do we still have time for the video, or do you think, what should we do about that?
Well, the video lasts about 3 minutes or so, so I think we could quickly show the video. John, do you think you can show that on your screen? Well, just give us a second. It's the first time we're showing a video on a webinar, so just give us a second and hopefully it'll come up.
So this is a video that we recorded up in Pennsylvania at Good Harvest Farms, and it basically just shows somebody carrying out the grafting procedure itself. Hopefully we can get it queued up here.
Okay, we should be able to see it any minute.
Maybe while we're waiting, I might talk about the dangers of grafting, and Carrie talked about sanitation. The other thing is sometimes when we have a good tool, we reuse it on the same land. So, of course, you need consistent rotation, or you might need to use different rootstocks over time.
Okay, yeah, so we see the video now, so if you just want to talk everyone through that.
Sure. So basically you can see Caitlyn here, and she's sort of introducing herself. One of the things that you can see is that she's about to carry out some proper sanitation methods. Caitlyn basically just uses a regular straight razor for carrying out the grafting procedure. So what we do to make sure they're nice and clean is just dip in a little bit of alcohol, and then you can just use a candle to flame them. Of course, it's really important to be very cautious when you're using any of these things. We're talking about knives and flames and alcohol, and so you should always be very careful. One of the things that's nice about what she's doing there is you can actually sanitize, 20 or 30 of these razor blades at a time, and then basically have those ready to go, so that you can see she has a pile of them on the right there as she's doing the grafting. That way she doesn't have to actually sanitize each one during the process. She does them ahead of time. Here you can see her doing the grafting, and this is what I was talking about cutting those strips into 20s. So basically you can work down one side, and she actually doesn't flip them around. She'll just cut it from the other side. These plants are a little bit on the large side. I would be a little bit cautious about these. They're sort of getting right at the top end of what we call a good size for grafting. But basically, one of the things that's nice about this tube grafting method is that it's real quick. And so you can see she's just quickly moving through these plants, and then she'll go ahead and put those clips on them. Here you can see her doing that. And those clips, too, they'll actually slide right over top of those stems. You don't necessarily have to come in from the side like that. They will slide right over top, and they easily go on there. You can see she's sort of adjusted the way she's doing this, basically so we can get a good camera view of it. But again, you can imagine how quickly you can just go ahead and put 10 clips on across the strip there. Now she'll go ahead and insert her Scion, and again, she's sort of awkwardly doing this because we're trying to film it and capture it by film. But you can see basically the stem goes right down in there, and you basically just want those angles to line up real nice and smooth. Now one of the things that I like to do when I'm actually grafting is I just be real consistent that I cut from the right to the left all the time, and that way I know which way my angle is going to be when it comes time to inserting the plants in. So for example, this person here would cut maybe from the right to the left. Then when she goes to the Scion and grabs the Scion, she would use that exact same angle, that same shape, and she'll know that the angle is going to fit when she brings it back to her rootstock. And this is what you want it to look like, is a nice, close, good. It's nice the way those angles really line up smoothly in that clip. And that's pretty much the whole process there. Now, once they go into the healing chamber, it's a different beast altogether. Again, managing that chamber can be very tricky because you need to keep the humidity up, but you want to keep the temperatures from getting extreme on you. And so that would be my highest recommendation, is just be real cautious when you're managing these things within the chamber. So if there's time, I think maybe we'll take a few questions. Alice or Frank?
Okay, yeah, I just wanted to repeat for those of you who joined the webinar a little bit later that if you would like to ask a question, all you do is you just type your question into the question box and hit return. And if you don't see the question box, you can click the small plus sign next to the word question to open it up. And, excuse me? Okay, yeah. We'll be reading the questions out loud, and we'll answer as many as we have time for, and we're just going to run a quick, like 20 to 30-second poll right before we start the question and answer period to just kind of measure how beneficial this webinar was to you in terms of increasing your understanding of how to graft tomatoes. So we'll just wait about 20 seconds. If you could just type in an answer, we'll move on. We've got a lot of good questions coming in. Okay, we'll just give it another few seconds, and yeah, then we'll move on. Okay, great, thank you. Okay, first question. We had a couple questions at the beginning of the webinar about self-grafting and why it works to reduce soil-borne diseases.
Yeah, I'll tackle that one. Carrie and I have seen that phenomena off and on, where sometimes self-grafting actually reduces soil-borne diseases, or sometimes we see decreased virus pressure. And what we think it relates to, and actually Carrie did some of this work in his thesis, is when you wound a plant, the plant will turn on some of its natural defense systems, either systemic acquired resistance or induced resistance systems. And so we're thinking that the wound response causes the plant to turn on these defense systems, and we see less disease. But we never see self-grafting work as well as a properly selected rootstock.
Okay. Another question. There was a listener wondering about whether or not you plant the rootstock first and what the lead time is between the time you plant the rootstock plant and the other plant that you're grafting onto it. Could you talk a little bit about that?
Sure, I can field that question, Alice. You know, one of the things that you'll find if you look to the internet for information about grafting is a lot of these things, including our own extension publication, will say plant the rootstock exactly 2 days ahead of the Scion or exactly 5 days ahead of the Scion. But bear in mind, all these things are going to germinate at slightly different speeds depending on what the greenhouse environment is that you're doing it. So the first thing I would recommend is when you get your rootstock seeds and your Scion seeds, take about 10 or 15 of these things and do a quick germination test and basically just grow the plants up to the grafting size and try and figure out how much you need to stagger those seeds before you start to plant 500 or 1,000 of these things. In general, I would say that a rootstock like Maxifort or Beaufort germinate at generally pretty much the same speed as most of our commercial hybrids. There have been a few cases where I've actually planted Maxifort after some of our heirlooms because our heirlooms may be a little bit slower. And there's sometimes where we plant rootstocks ahead of time. So in the case of RSTO 4105, that's a very slow-growing rootstock, and we'll plant that one 4 or 5 days ahead of any of our commercials for the Scion. So it's really going to vary on your rootstock and your Scion combination as well as your environment. And so again, the most important thing to do is just do a test. You know, try and figure that out on your own, and you'll have a lot better luck that way.
Okay, here's another question. With the callus formed at the graft union, can the grafted plants be mechanically transplanted and handled like non-grafted plant at time of setting in the field?
Overall, I would say yes. That graft union is actually quite strong, and we handle them just like other transplants. Carrie, do you have any information on where people have actually used a transplanter?
You know, I haven't really seen a mechanical transplanter run with grafted plants offhand, but I would echo what you just said. Once we harden them off, like we do any other non-grafted plant, we have no problem with these things falling apart in the field. They're real rigid. It's never.
My gut feeling would be if you have one of the more modern carousel planters, then that would work well. I could see some danger with the old Holland transplanter that has the fingers, and you probably need to be gentler with that because I can see when you use the old Holland transplanters, you place your fingers on those stems, and if you put too much pressure, I can see how you can crack it. So just offhand, I would say there might be less danger with a carousel transplanter. But in our experience, those transplants, we treat them just as roughly or as kindly as regular transplants with no problem.
And I treat plants pretty roughly. So.
I'm
Okay.
a gentle type.
We have one more question about transplants, and that would be the tray cell size. How small or volume of soil is reasonable?
Sure, I can answer that one. One of the things that I found pretty quickly is that, you know, growing up in the greenhouse business, I like to use those big, deep 606s or a 36 cell tray that makes a nice, hardy plant, stocky plant. But keep in mind, you're going to have to work on the stem of this plant, and so you actually want it to grow a little bit taller. And I wouldn't use the word lanky at all. If you're getting to a lanky plant, then that's bad. But you want them to have kind of at a high planting density so that you stretch out that stem a little bit to carry out the grafting process. So we've had a lot of great luck with a 50 cell tray. The 36 cell tray, again, it widens them out a little bit too much and the plants are a little too squatty. 50 cell tray works good. I would think a 72 cell tray would also work very well. If you get much higher density than that, they're going to start to get lanky. And I've actually seen, you know, these grafting robots. They use plants grown in 288 cell trays, which are really, really, really stretched out. And one of the things that can be a problem is then when you try and put it back into the field, you know, you may have a problem with that plant just being a little bit too wimpy to survive the wind. So I would stick with a 50 or 72 cell tray.
Okay, great. Can you tell us what temperature and humidity you need in a healing chamber?
Sure. So the humidity is going to vary, of course, but in the first day, you want it to be as high as you can get it, like above 90%. You know, it's hard to get hygrometers that measure it actually that high, but you know, if you use one of these vaporizers or something like that, it's going to pump it up really high. Probably about the 5th day in the healing chamber, you'll want to start cutting back that humidity. And so one of the ways we do this is we can either vent the chamber or we'll actually turn the vaporizer down, which is a really easy way to do that as well. In terms of temperature, you know, you basically want it optimum growth temperature for tomatoes. Keep in mind that if it gets too hot, they're gonna cook in there. I like to keep them, you know, if I can keep them around 75 to 85 inside of that healing chamber, I'm doing really well. It's hard to keep it from getting, you know, too warm. Definitely don't want it above 95 inside of that chamber, though. That's probably too hot. There's actually still a lot of research to be done to describe some of these specific numbers because a lot of it we sort of just been figuring out how to do without collecting enough data along the way. So we're hoping to continue some research in this area as well, too.
Okay, we have a listener from Illinois who is wondering if there have been any tests with early and late blight resistance and grafting.
You know, that's a great question, and there's a twofold answer. One, I would say for organic growers, you know, in North Carolina, early blight is our most troubling nemesis. We get it every year. We get lots of rain. Really, you need a Scion that has resistance to early blight and late blight. And there are some breeding programs. I know we have one here in the mountains where they're actually heirloom quality tomatoes that have early blight resistance and late blight resistance. And that's going to be the best approach. Now, sometimes we see with certain rootstocks, we do see decreased foliar incidence. And there is some data in the literature, not too much for early blight and late blight. But what happens is if you have a better growing plant and you delay early senescence or early death, which some of these rootstocks do, then you do tend to get less early blight. I don't know about late blight. Late blight likes green, young tissue. And when it blows in, there is nothing you can do, you know, unfortunately. So the twofold answer is yes, with rootstocks, sometimes we do see a delay in the amount of disease, but to really control it, you need to select Scions that have resistance. And there are some coming down the pipeline.
Great. Okay, we have two questions that are basically the same question. In your studies looking at the effect of grafting on disease control, did you inoculate the soil with the pathogen? And also, besides soil-borne diseases, did you test any other diseases?
Well, you see, this is the joy of living in the South and working with some of these organic growers, is we didn't have to inoculate our soils. We have a lot of very good disease pressure. So all those were in fields that had natural inoculum levels, and most of those were on farms. And so like that one farm, you know, we went from 100% plant death to 100% plant stand, and grafting functionally put that grower back into the tomato business. So yes, so we did not, we relied on natural inoculum for all those cases.
Okay. Let's see, is it better to grow grafted tomatoes in a greenhouse versus outside?
Gary, you want to tackle that one?
Sure, you in terms of production, or is that what
Well,
we're talking about?
Yeah, I think so. Probably, like, where's the most benefit, do you think, that you'll see?
Yeah, so you know that's one of the things that we tried to get at with that research that we did at the Center for Environmental Farming Systems is, you know, comparing the benefit of grafting indoors versus outdoors. In that case, we actually found that it was just as beneficial inside as it was outside. Now, one of the reasons I think for that, just intuitively, is because we're using Cherokee Purple for our Scion, and Cherokee Purple loves to be grafted. In almost all of the trials where we used that as a Scion, we found significant yield effects. In fact, I think in all of the trials where we used that Scion, we had significant yield effects. I would say if you're using a really vigorous heirloom Scion like German Johnson or some of these other indeterminates, you may not necessarily see a benefit in the open field. We've seen that happen as well too. And you know, one of the things that has sort of been assumed about this whole grafting thing is that we get a lot of added vigor in the tail end of the season, and so maybe the longer the growing season, maybe the more benefit you would get, and that's sort of one of the things that we expected to see with the CEFS research. So we were actually really surprised to find out that in the case of Cherokee Purple, in fact, we saw benefit sort of early to midway through the season, and that in addition, it was the same in the field as it was indoors. But I think in general, you might see more benefit in the greenhouse than you would in the open field.
Yeah, and I would say it depends on your production system. Like 45% of our tomatoes in the grocery store now come from greenhouse operations, and all those plants are grafted. It's about 40 million plants, it's estimated. And so those plants are grown in hydroponic systems for 10 months or more. And so grafting has a very distinct benefit in that system. So, you know, that last statement Carrie made, that if you have a tomato greenhouse production system where you extend the life of the plant and you can train twin leaders, grafting is used almost exclusively now.
And that's right. And in the field, you know, we think it may be helpful, but you know, it's also important to just maybe try it in small numbers before you start committing 100 acres or anything like that. So.
We have a question about whether the rootstocks are all hybrids or whether any open pollinated varieties are used as rootstocks.
Yeah, so in my section there, I talked about CRA 66 and Hawaii 7998, and those are open pollinated lines. They're actually breeding lines. One's from India originally, and the other one, I guess maybe it's a selection in Hawaii or something like that, but they are commonly used as rootstocks for bacterial wilt control, and they work very effectively. And so those are open pollinated, so a grower, if they got seed, they could produce their own seed if they have the right sanitation measures. Otherwise, most of the rootstocks that are commercially available will be hybrids, and therefore they have that price tag also.
Okay. Another practical question. How much leaf should you remove on the Scion?
Yeah, that's a great question. Carrie, I'll let you tackle that one.
Well, generally we just do de-leafing to help with air circulation and pruning. We like to prune up to the first fruit hand,
No, but this, I think this is in the grafting procedure itself. Like in the Scion, when you're actually doing the grafting, will you prune away leaves? That's my understanding of the question.
I see what you mean. I typically don't prune away any leaves. That's something, we're kind of playing around with that area right now. But we typically have not pruned any leaves in the past.
And if you look at some of those links that we provided on that handout, Ohio, you'll see they use a cleft graft procedure. Cleft grafting has a little more tolerance for differences in sizes of the stems. And then they also will tend to prune the leaves more than what we've done. And so that would decrease the transpiration stress on the plant.
Okay. Speaking of the handout will be available later today on the website, a link that I just sent along. It's www.extension.org/article 32969. So immediately following the webinar, I will make that available. I thought it was already there, but it actually isn't. So yeah, we will make that available right away after the webinar so that you can go to that link and then you can find all these resources. Okay, time for several more questions. Do you recommend grafting for determinate tomatoes? And if so, what rootstock and what is the yield increase?
Yeah, we've seen good responses for both determinate and indeterminate types. And again, it depends on the situation. When we have severe disease pressure, then we choose the rootstock that suits that site. So if we have bacterial wilt, we need bacterial wilt resistant rootstock. If we have nematode pressure, we need rootstock that has the MI gene. And so, especially under those conditions, we've seen good yield responses. We've had cases where we've had grafting and everything was optimal and we don't see a yield benefit in fields with no obvious disease pressure. I can recall one case with an organic grower, and it was just a very optimal production system. We didn't see a dramatic benefit. In contrast, we showed the data from those high tunnels and open fields in eastern North Carolina. Even in the absence of disease pressure, we did see a benefit. So, it's a good scientific question. I mean, the answer is a very scientific answer. It depends. So it depends on if there's disease pressure or not. That would help you determine the rootstock selection. And it depends what kind of stress is on that plant. And, the rootstocks seem to give greater abiotic, antibiotic stress. And then we do see a yield response. Carrie, you want to relate your experiences?
In terms of determinate tomatoes, yeah, we've had great experience. One of the things that I think is nice about using determinates for grafting is a lot of these rootstocks that we graft onto, for instance, Maxifort, it tends to make the plant a little bit more vegetative and maybe grow a little bit more vigorously than we like. Especially some of these heirlooms, they may get really tall and hard to manage, where the determinates will have bigger plants, but it seems like the determinate Scion is better at adjusting that energy into fruit production. And so we've always had great success in terms of yield effects using determinates. So yeah, I wouldn't hesitate to graft a determinate at all.
Okay, we have a question about grafting clips. The person thought he heard you say that the clips were 2 millimeters, but the movie looked like they're 2 centimeters. I think 2 centimeters is correct. Is that right? Or
they're 2 centimeters tall, but the inside of that stem diameter is actually 2 millimeters. Yeah, and they come in 2 different sizes. There's 1.5 and 2. Probably 98% of the grafting we did was with a 2 millimeter size, but I like to keep 1.5s around as well.
Okay, another couple of questions. We had a couple of questions about Beaufort. One person wanted to know a little bit more about what that is, and then another questioner wanted to know whether you found that Beaufort is easier to manage in the field and has slightly less vigor.
Yeah, that's I think there's some seed catalogs that have said that. And we've seen pretty similar trends in the field. Beaufort is not as vegetative as Maxifort, and so it's typically recommended for heirloom varieties, especially these indeterminate varieties. We haven't used it a lot with our determinates. We typically will graft our determinates with Maxifort, but that was sort of the context of the studies we were doing was driving that decision more than anything. But overall, Beaufort has done very well in our field trials, and comparable with its sister, Maxifort, you know, they say Maxifort's more vigorous, but where we see significant yield effects with Maxifort, we generally see them with Beaufort as well. So, you know, it's a good rootstock, and it's always done well for us. And it also does seem to show a little bit less of that generative growth, which is nice for trying to manage those heirlooms in particular.
Okay, we have quite a few more questions coming in. We only have time for a couple more. Let's see. Is there a problem with burying the graft union?
Yeah, that's...
Go ahead, Frank.
Okay, yeah, because what happens, tomatoes are very good at putting out adventitious roots. Once you bury that union, then the top will produce its own roots. Once those roots touch the soil and pick up the pathogen, then it will get disease. So you need to make sure that no Scion roots will grow into the soil.
Okay. Let's see, here's an interesting question. Do the grafted and non-grafted tomatoes taste the same?
Hey, Carrie, you're the taste tester, I think, huh?
I haven't been able to taste that myself or test that myself. I usually just throw them all in a bucket and they get eaten at the end of the day, to be honest with you. But I think that you mentioned they're doing some of that work down in Florida, Frank. Can you speak to that?
Yeah, there's a lot of work on that, you know, where people are looking for two things, texture, flavor, and also health benefits like lycopene antioxidant content. I would say so far, from what I've seen in the data with tomatoes, there are not dramatic differences. But the expectation is that as grafting matures and we enhance the rootstocks, maybe you can select the rootstocks that do enhance taste or fruit quality. Now, if you're going to graft watermelons and melons, sometimes they do see dramatic fruit quality effects in taste, texture, and health benefits. So, you know, depending on the crop. But overall in tomatoes, I would say we have not seen that in the data. And you know, I don't know the data intimately at Ohio and Florida, but that would be my read of it.
Okay. Well, we are running out of time, but I'd like to thank you for all of your questions. And as I mentioned before, if you have additional questions, you can use the Ask an Expert system at extension.org/ask. This webinar will also be posted on the eOrganic website at extension.org/organic_production under our archived webinars. That's probably the easiest way to find it, or you can just Google the title of the webinar, Grafting for Disease Management in Organic Tomato Production Webinar, and you should be able to find it. And probably within the next 10 minutes or so, that resource handout will be available. I just republished that, so that will be available, and the slides from the webinar will also be there. So if you wanted to see anything again, you'll be able to look at those. Thank you so much for coming again today, Frank and Carrie, and thank you all for joining us.
It's been great. Really enjoyed it.
Yep,
Thank you.
Thank you very much.